Okay, see Jesse left a message. He won't be able to speak too much today, but that's fine. Sushmanth, hello? Wait for some more people to show up. Does anyone have any updates or anything they'd like to give? Or hello.
Good morning.
Morning. How are you?
Sleepy.
Yeah. All right. So, did we have any updates on anything before we get started?
Nothing. Do you have time afterwards to finish the paper?
Yeah. What do we need for that?
We need to discuss a few minor points.
Okay.
Okay.
We can do that now, I guess.
Okay. Things like are K and N identical?
Oh.
K and lowercase n.
Yeah.
I changed all of Bashir's equations to K.
Okay.
Okay.
All right.
And the other question you asked, you had mixed proportional and Poisson distributions.
Yeah.
And you said that's unique with you, but I thought it might be in the radiology literature.
Oh, okay. Well, I'll look for that. Yeah.
Okay, those are the two points I recall as being outstanding.
All right. I don't know. Yeah. So Bashir changed his equations, or?
No, he fixed them.
Okay.
He fixed them because somehow when they went through your machine, they got screwed up.
Oh.
I fixed them and then he double checked.
Okay.
Okay.
Yeah. All right. Well, I'll take a look. Yeah, we'll talk about that. If you want to talk about that later, we can.
Yeah, I'll switch computers at that time too because that's on the other computer.
Oh, okay. Yeah.
Okay.
All right.
Okay, that's it.
All right, that's it. Jesse said, morning, just mostly working on going through the A-Life submissions and grad work. I'm going to be busy 2 weeks for work and maybe on pause until then. Okay, that's fine. Thanks for the update. Sushmanth, did you have any updates?
Yeah, actually, I have in the last couple of days.
Oh, it's dark there.
Oh, yeah.
We have lots of sunlight and still lots of snow.
Okay.
Yeah, I'm going through that tool actually. It is quite interesting and easy to use. And there is a separate repo for the cell division and morphogenesis actually. I'm going through the whole thing. And what is your idea on the I haven't got any clear idea on what you have been telling on that day on the Slack channel. Can you just elaborate it, Bradley, if it's possible?
What was this for? I think you cut out earlier.
That instance segmentation. You are asking about to make a module of DevoLearn. I have discussed with Mayukdeb actually. He told we can make it, but I didn't get you what was there in your mind clearly. Can you just explain if it's possible?
Yeah, so let me explain. Let's see, I can share my screen. I'll just draw it out and we'll talk about it. But the idea was that, so we've been wanting to do a lot of different things here with the software eventually. One thing would be to get single cells and segment those. Another would be if you have a lineage tree, like we have in C. elegans, where it's well defined for each cell, to map that to it. Then we have, tissues and other types of things that you can extract from embryos that are more developed, that have tissues and things, and be able to take those and use semantic segmentation to segment out parts of the tissue and, things like that. So we want to do things at a number of different levels. But one thing that would be nice is we could, because now we're kind of, diversifying how we're doing the DevoLearn aspect, is to have different modules that would do different things. So, under DevoLearn, we have right now So we have semantic segmentation.
Yeah.
And this is part of this pre-trained model, right?
Yeah.
And so this is kind of the origins of this. And so that comes from like this basic need for segmenting cells. So you segment images. And then the training data, is it could be images, or microscopy. It could be text, or I guess we're using for lineage trees would be text or trees, like graphical structures even, because you could take like a network or a tree, which is a graphical structure, and you can put it into, you can train it with that. So, a lot of the microscopy data sets will have like a bunch of cells. And, you can segment the cells, like this is just an area, just a generic area of cells. And you want to segment out these cells, as independent units with centroids. But sometimes you don't have the identities, like in mouse embryo, for example. Each cell has depending on the mouse, depending on the sample, these cells have a different identity.
In the first 30 minutes.
So the lineage tree is, it changes depending on the mouse. So this has been a problem in reconstructing lineage trees. So we don't really want to connect explicitly some of the like graphical data or some of the label data to the cells directly. We want to be able to be flexible. So one thing we can do, of course, is have the semantic segmentation, but that's not always going to help us. Then we want to have maybe instance segmentation. So this would be for things, embryos or images where we don't know the identity of the components, which is sort of the problem that you've been working on. And you notice that, like, it's not going to give you a great resolution, but it depends on what algorithm you use. It depends on the images. So we've had problems in the past with different types of images where the images are more or less amenable to segmentation. So in the past, we've tried different things, like, you'll have bright field, which is a type of imaging, which is where you have like just the backlit, a backlit slide with cells on them. It looks grayscale. Those are not really amenable to a lot of methods that we've used. They use things like ImageJ, which is a program where they can, have to transform the image and get it to like a different color palette to augment the edges. What we've done for machine learning is we've augmented the edges using different augmentation techniques. So, under bright field, you have augmentation techniques, which are like basically highlighting the edges or the features you want to extract. So in past years, we've had people do things like, highlight the edges of the membranes with contrast, or done it, sometimes manually. That's not really that's not a really tractable method, because, you can't imagine, I couldn't imagine someone going through 1,000 images and doing that. You could do it, but it's not efficient. So we want to move beyond that and try to get, other types of images. We've got fluorescent images, which actually have the benefit there. Yeah, fluorescent images. So the benefit there is you have those kind of things marked with a fluorescent marker. So sometimes people will generate data with like the membrane as some sort of fluorescent marker, or the nucleus with a fluorescent marker. And the problem there is that, the benefit of that is you have a pretty easy hook on which to do your segmentation. I mean, all you have to do is transform it to a slightly different color palette, and you can get a binary representation of the boundaries. So it would be pretty easy. The downside of that is the fluorescence. Sometimes it autofluoresces. Sometimes it's hard to get the things above the threshold. So, you have breaks in the membranes or breaks in the fluorescent signal spatially on the image. And so, you'll have like maybe some lines like this, and there'll be breaks because the fluorescent signal isn't strong enough to show. And so this would be the boundary of a cell, and you'd have to like fill that in. But some, the algorithm may or may not do that. So you have those kind of problems. And I think that may be some of the problem in the data that we have with, some of the data sets like the EPIC data set, where you have this kind of thing where the fluorescence is uneven for different structures. But that's a problem that we have had. It's a common thing we've had to do in the past. So that's like something that it's hard to really kind of find the killer algorithm to fix that. It's just a feature of the data set. So that then there's this pre-processing that one can do to fix that, with playing around with the color palettes and how the image is represented before you actually put it into the algorithm. That can be useful, but not not fully solve the problem. And then we have types of images. We have SPIM imaging, which is something we've been using with C. elegans. There are these high resolution microscopy images that it's a type of microscopy where it's very high resolution. It kind of looks like bright field imaging, but it's not. It's a different method. And that gives you probably the best resolution. And I have a folder of different images that we've used in the past, sort of our stock images, if you will. And those have a lot of these images in it. I'll probably put that out soon. I didn't think about that when we were talking about this. We're just getting started on it, but so the EPIC data set is good for kind of like bulk training. And the bulk training...
Actually, EPIC data set has become a bit old.
I know.
Not the problem. Yeah. And we can follow up with cell tracking challenge data set actually. They have released they have like all type of cells, mouse cells, CL GAN, B-Mellings, etc. But the cell tracking challenge data set is pretty good. I have gone through like some of the files from it. They are pretty we can train our model with that, and it's good. What your thoughts on instance segmentation? You're okay with my idea, right?
Oh yeah.
I just have small doubt. I talked with Mayuk. I told him like DevoGraph stage 1 needs centroids and volume of each cell. So he was like, I shown him the segmentation results actually. I think he's like, he want to join today's meeting, but due to with some other meeting classes, he haven't joined. He was like pretty showing interest, like we can make the segmentation models. We can extract centroid of each cell and volume of each cell. Actually, right now, DevoLearn situation is like this. It can't extract the volume of each cell. But in DevoGraph stage 1, we need volume of each cell also. So we need to see what's your thought on my idea, like instance segmentation?
Yeah. So we have we've tried to approximate volume in different ways, and it's hard to approximate volume right now in DevoLearn. So I think we definitely should have that in the pipeline. And so but what my point was in like a lot of the stuff we've been talking about is that I wanted to keep them kind of separate into separate different modules, and then in the documentation we can bring them together. So like, if I want to do something with this at the end of the day, what do I want to do? Like, if I want to... Oh, what was that?
Yeah, go on. Sorry.
Okay. So at the end of the day, what do I want to do? If I want to do something with this data, with DevoLearn. So say I want to like create a situation where I have an embryo and I have different cells. I'm just segmenting the embryo into different say I want to do the segments here, right? And these little parts are not cells, they're just kind of like regions. And I want to create these regions so that I can like look within them. So for the regions, I might use semantic segmentation. All right. And then that just defines my regions. So region A, and I want to put a label on it. and I have the labels, and then that may be a tissue, that might be something that we track over time. Now within that, maybe I want to know what are the cells. So I'm tracking it over time. You have all these cells, you have like some founder cells, and then they divide and they give you a bunch of cells in there. Now we don't have the labels for this, right? Let's just assume.
Yeah.
So we can't really use that. So now this is where you would use instance segmentation.
Yeah.
All right. Now we want to be able to put together something where someone can take this thing, this tool, and say, I want to look at the cells in this embryo. So I want to use an instance segmentation algorithm or the toolbox. And then I want to find maybe something out about this region. So we'll use the semantic segmentation toolbox. And then, I don't know, maybe like for the graph neural networks, you want to put these together as a graph where you can connect the cells or connect these regions according to different criteria. So then there's this graphical representation. And this would also work for like a lineage tree or a differentiation tree, which we've talked about, or anything else where there's a graphical representation. So that's maybe how we want to do it, because I want to make this flexible, and there are a lot of things in development you'll need these different methods for. So that's my thinking, is that we could have like a toolbox for each of these. We've got a lot of the semantic stuff. We have the instance. You're gonna propose that we put the instance stuff in there, and then the graphical stuff would be sort of like the GNN's work. And so just kind of improving upon all of these would be good. And then we'd have you could choose what you want to use for the task you want to do. So if it's like instance segmentation or a semantic, whatever you want to do, whatever the task is, you don't have to use this one generic thing to do it. You can use these different things.
Bradley,
Yeah.
could you go back to your previous sketch?
Oh, this one here? We're back.
Back. Yeah. As you may recall, my mother did one picture of Drosophila fixing up the segmentation. And I think it took her somewhere between 10 and 100 hours.
Yeah.
For one picture.
Yeah.
But we never developed any tools to help her. Okay, and I was looking at that and realized that that's possible. That a tool that is a mixture of a hand and automatic might work.
Yeah.
Okay, now let me give you an example. So look at your sketch that says epic.
Yeah, right here.
You could determine the centroid for those segments.
Right.
Okay, and then from the centroid, you could send out rays in all directions, and you could indicate when they hit something. Now, those that are near it will hit it sooner, and those where there's a gap, they'll go out and hit something else. So if you looked at the distribution of the lengths of the rays, you could select for those that belong to the cell.
Right.
And then you could simply draw straight lines to connect their edges or curves.
Great.
Bradley, can I show one example? Actually, I have a segmentation result of instance segmentation. I would love to show to you.
Yeah.
Sure.
Yeah.
See a real example.
Yeah. Okay, go ahead.
Yeah. Actually, this is a picture from cell tracking data set. Actually,
Okay.
see, this is like fluorescence image, microscopic image from cell tracking challenge data set. These are mouse cells. I have trained the model on mouse cells, and I tried to segment this one. These are the segmentation results. Actually, I hope you can see this.
Oh yeah, it's great.
I mean, every cell is actually properly segmenting, except a back one. This cell is not segmented. Other cells are completely cleanly segmenting. You can see these results. From this, I was thinking to extract the fuzzy. Actually, my network has 2 days back has told me you told one 3D tool to Fiji, F-I, I will keep a spelling of it, F-I-J-I, Fiji, an open source platform for plotting in,
Oh yeah, Fiji. Yeah.
yeah. With that, we can, by using that tool, we can extract the centroids of these cells in 3D positions, and volume of cells also we can calculate. We can calculate by using OpenCV library, we can calculate the area of each cell. After that, how many frames, like video frames it is there, we can extract the volume of each cell. This is the idea of extracting it. Actually, I was talking with Mayuk and Mainak. I had a meeting with them. They are like, okay, these after seeing these segmentation results, they told like, yeah, you can make this out. They have given a green signal, like, yeah, you can do it. I mean, these are the segmentation results with the code I have right now. I can train it more and extract for C elegans also. And actually, I have the results of these are C elegans. I just tried to extract for C elegans for one frame, then I got these results. This is like 128 cell stage actually. These are results of C elegans. This is for mouse cell.
Yeah, those look great. So yeah, the cell tracking challenge, what kind of data is that? Like what organism and what type of cell is it?
Actually, it contains all type of organisms. C elegans, mouse track. I mean, I will share the link. Wait a minute. You can see everything is there. Every cells are there in that data set. It's not particularly for one.
Right. That's the
time data set. 3D data sets are also there. We can use it. I'm sharing them. Yeah, I have put the link actually. You can see the data sets. Actually, DevoLearn was trained on this data. 2D data set. When they are trying to, actually, with me, it was mentioned cell tracking. By using cell tracking only, they have trained the DevoLearn model.
Yeah, I see that. Yeah, I remember the Waterston lab data. We've used that in years past, and then there are other types of data like for cancer cells. Those are usually always available, like in some form. Yeah, there's a lot of good stuff in here. There are other types of embryos like the Trilobite castaneum embryo. There's there are all sorts of different types of cells here.
We can train our model, and we can extract for demailing star also, because demailing star will go up to 2,500 cells. Before writing code, I have gone through the data sets actually.
Yeah.
And I understood like what happens in. I am not good at spellings. Demailing star. I hope that's the spelling of it.
Oh, what was that?
Demailing star. I don't get the spelling.
Yeah.
Really, I'm not good with names. Sorry.
That's okay.
Yeah, I have analyzed the cells, like how they divide. It was going up till 2,500 cells. At that time, also, whatever the instant segmentation I have done, it is working fine. And it is working fine for mouse embryo cells and even for C elegans. So we can implement this idea. I'm attaching the results in my proposal also, like what I was getting. And the main work would be like segmenting more, like training it on the data of C elegans first, and after that extracting the centroids on time series data and the volume of each cell. That's what I want to do. And with that attaching, I was trying to do topological data, and the tool which you have shared 2 days back, it's quite good. We can do it with that.
Yeah.
That's what is going on with me.
Well, that's great. Yeah.
Bradly?
Yes.
Oh, yeah.
Yeah, Bradly, getting back to sketch, I realized I didn't explain it properly. The centroid will work even if it's just guessed at by touching the screen.
Okay. Yeah.
You see, this method will work independently of whether you know the real centroid.
Right.
Okay.
Yeah. Yeah, isn't that kind of like the breast cancer stuff that or just the physical?
No.
Okay.
No, it's It's actually similar, somewhat similar to the stuff I did on, what do you call it, adaptive neighborhoods.
Oh, okay.
You take a point and you get a distribution of something around the point and use that to characterize the point.
All right.
In adaptive neighborhoods. Here what you're doing is taking rays from a point inside that group of line segments, and you're characterizing the length of the rays from that point, distribution of them, and saying, okay, the cell has got to be amongst the closest ones. So you pick the closest ones, and then you can close them off.
Okay.
Okay, and since they're arc segments, it's easy to close them off consecutively and get a proper image of the cell.
Yeah.
So yeah, I think you have to think more like how we think. I mean, we spot these discrepancies very fast.
Yeah.
Okay, so we have to sort of simulate our own thinking process.
Right. Human intuition, basically.
Yeah, human intuition, turning human intuition into code.
Yeah.
Okay, so I'm saying we shouldn't give up on data sets that are imperfect, but develop perhaps a technique for rapidly making them perfect.
Right. Yeah.
Okay?
Yeah.
And you see, all you have to do is point to each cell that looks defective, and then the algorithm would fix it.
Right.
Okay, that's what I meant by a mixed human-machine approach.
Right.
And that would be much faster than drawing all those little line segments.
Yeah. Yeah.
Okay, and perhaps more accurate.
Yeah. 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 The trick is getting it to work like perfect, or getting it to work flawlessly.
Well,
Yeah.
we certainly have data sets to test it on, so
Yeah. Yeah.
go back to the original Drosophila set and see if we can do it semi-automatically.
Oh, right. Yeah.
Because it was, so to speak, full of holes.
Yeah. Yeah. I remember that.
Yes.
Closing, yeah. So even though when that got digitized, like, when we were doing because it was a drawing that was,
Yeah.
the drawing was, the loops were closed on that, but then when that got segmented, there was some work that needed to be done additionally to the digital.
Yeah, it was a big job.
Yeah. But that would be to be able to just say, okay, this is basically what we want, and then here's the intuition, and then moving from that to the actual.
Yes.
Right.
Yes. It's I think it's what's called filling in.
Right. Right.
In terms of vision.
Yeah, it's like when the visual system.
The visual system fills in very well.
Yeah.
In fact, deceptively so.
Yeah. Yeah. And yeah, sometimes it is deceptive, but yeah, but we can usually know, like, well, we can't necessarily if we have like line segments that are like, they did these experiments with line segments, and they ask, are these the same length or different lengths? And they draw.
Well, you need to look at the distribution of lengths.
Yeah.
And then say, okay, I got a peak of lengths here that are shorter ones, so I'm going to assume those are correct.
Right.
You could even possibly automate the whole process by finding the centroids of the segments.
Yeah.
Okay.
Yeah.
That's not out of the question, because your eye will probably rapidly find a cell which is not completely closed.
Yeah.
And you can imagine an algorithm that could rapidly find a line that ends and doesn't close.
Right.
I mean, if it closes, you start at that point, you go around the whole group, and you come back to where you are. If it doesn't close, it doesn't close. Okay.
All right.
So there may be ways to do the whole thing automatically, but in any case, tools for rescuing bad data sets are perhaps warranted.
Right. Yeah. I don't know, they're bad data sets, they're just bad for what we want to do.
Well, okay.
Yeah.
Okay.
Well,
Yes.
yeah, getting them digitized basically.
Yeah.
So yeah, that's what we had for we have a lot of potential microscopy data sets, whatever we want to use. So, and then there are things out in the public domain that we can use that, I don't know what the most recent data sets are that people are using, but a lot of the C. elegans ones are a bit old. I think they started really doing some of this about 15 years ago, and like I said, some of the data sets are better than others for certain things. So it's like really finding.
Yeah, if we go back to the Basilaria, there were some where it was difficult to segment one cell from another. But if you could manually identify the center of a cell and put in the information on the maximum width of the cell and maximum length, then you could probably segment two cells that are difficult to segment.
Right. Yeah, I think that's what we were doing, like with some of the images, like we were drawing bounding boxes and getting different, like using it as a training module, like a training.
Okay.
Yeah, so it's like a template, and then you would apply it across the different cells to say this is basically a cell. Where do you see the end of the cell? The sides of the cell. You could detect different parts of it, and then just put the bounding box around it.
Did that work even if the cell was badly segmented from its adjacent cell?
For the most part, there were some issues with that. I mean, you would get things where it wouldn't fully segment, and then there was some refinement that needed to be done from those. Okay. So, but it was and it was just a square, so it wasn't or a rectangle.
Or a rectangle, yeah.
So it wasn't like the actual cell, which is more of a oblong shape, like a cigar almost. Yeah. So I don't know if that, in practical sense, I don't know if that makes any difference. I guess it does, but yeah. The point of the Basilaria simulations was to simulate movement because they're moving, and that's what we were interested in. Yeah. So you also have time information. Right.
Well, we had it also here in Sushmanth's data.
Yeah. And so, in that case, we didn't really care about the division of the cells. We cared about they were moving against one another. So I know Sushmanth probably doesn't know this work, but it's where we had these colonies of cells, and we would model each cell. They kind of slide against one another in a colony. We were interested in the movement of the cells against one another, but we'd had to model the cell body and sort of the boundaries and the edges relative to one another. So, we just drew these bounding boxes to define the cell length and the width, and then, they're adjacent to one another, and we'd observe these over time so we could track them. And it was actually a pretty good heuristic because, you basically were able to get that space, those two things together sort of next to one another, and you could get the entire colony and just characterize the movement from that. Because you could get the centroid, you could get the displacement, and other variables.
Okay. Yeah, I guess I'm just suggesting something. You start by guessing at the centroid.
Right. Yeah. Yeah.
Okay. You don't have to get the exact centroid.
Yeah. So the centroid is always like, it's an educated guess anyways. Like if we don't have the nucleus data, sometimes the nucleus isn't exactly in the center of the cell, but the reason we'd use a centroid in this case would be to say, this is maybe like the center of mass or the center of gravity, or it's like the center spatially. So like this is the center point of all the parts of the edges. And then we can say, okay, this is like the mean center of this thing. And then there's another thing over here, So it fits into not only the segmentation goal, but the graphical goal of like finding distances or finding an edge in a network,
So even if the edge between the adjacent cells is blurred, you might be able to work it out.
Yeah. So but that's But
Not clear idea actually, like how to implement and all. So thanks for it.
oh yeah, that's
In proposal, I think last time when you read my proposal, Bradley, you read the instance segmentation. Like, if possible, I have made some changes right now, like how I would segment and everything is it, how DevoLearn is there, how DevoLearn will be, like my idea of segmentation will be look. I have like so many, like before writing proposal, I have gone through data sets actually, some research on instance segmentation. After getting a clear idea on that, wrote the proposal. I'm adding a bit how I will implement topological data analysis also. And after that, please give a look to the proposal and let me know if possible.
okay. Yeah, I will. Yeah, I'll take a look again. And as for topological data analysis, it's like I have some references and I haven't put them out. But I mean, like the problem is that like it's depending on the problem, there are a lot of ways you can approach it. So basically, topological data analysis is where you get these patterns in the data. They're spatial patterns, and then you basically want to find different patterns. So, for example, if you wanted to find like toroids or different types of shapes in the data, you could use that as a way to that would be like a topological data analysis where you'd find like a shape in the data, like in the data cloud, basically. It's a way to do that. It's kind of a mix of like spatial analysis and sort of shape analysis. And depending on your data, it'll give you some really unique insights. But yeah, I mean, I can give you some general information about it, but it's kind of hard to, there are a lot of ways people do this. There's no real standard way of doing it. So that's the thing we have to keep in mind there. It's just
actually this link helped me a lot. I'm sharing in the this is for like CTA tool for morphogenesis.
Oh, this is nice.
Actually, this tool was shared by you only. After going through whole repo, I understood like this is what we needed for our project.
Yeah, this looks really good.
They have the like we need to implement this on cell tracking challenge dataset, and then we will get whatever data analysis we want, like centroids and the boundaries of it, 3D image segmentation, like showed in this video. So that's it. I will add some changes to proposal and I will let you.
Yeah, that would be great. Yeah. So let me share my screen here with people. We can look at this. So this is a resource on, let's see. This is a resource on this software, but it has like a lot of things for topological data analysis. So you can do things like manifold learning, which is where you find shapes within the data. So you're looking at, you're looking at a point cloud and you're looking at these different shapes within the point cloud. This is, I think, a general example. So it's not really like, it's kind of a toy example. It's not really anything challenging, but they just want to demonstrate it. There are other types of things, like different instabilities that you might want to find in a flow. You have contour trees. You have, merge tree clustering. So this is another way you can look at flows and things like that. Persistence clustering, which is, I can't remember what persistence is in this case, but I think it's basically things that exist over time. And yeah, so you have they have these different methods for that. Persistence clustering gallery, so you can see some of these shapes that they've extracted. So it's like, you have a point cloud and you don't know really how to interpret it. You can find different shapes in there in different ways. So they have algorithms here. You have time tracking, which is tracking over time of different things. Like in a flow, you have something that starts at an initial condition and then it unfolds over time and you can track that. You have tectonic puzzles, which are like when you look at the Earth and its tectonic plates, can you segment it according to the tectonic plates? So this is a geophysical model, but I don't know if you could use this on a sphere, a general sphere.
Paste it right into the email message.
So yeah, there are a lot of nice tools here.
I didn't check his.
So
Just paste it. Just copy and paste it. Stop attaching docx.
okay, yeah. So that's great. That's great stuff. I think it was useful to go through that because I every year I like to go through like what we have, and this will be useful for people if you're applying to GSoC for this project, and like the project that's listed in the NeuroStars. This is basically a project that is sort of maintenance oriented. We want to upgrade some of the things that we have in place. So, Sushmanth is working in his project, and there are other opportunities as well to do other things in the project. There's the graph neural networks pipeline. There's this part where we're doing segmentation to get to the graph neural networks. There's the topological data analysis. So there will be all sorts of things people can propose. And if you're applying to the project or you're applying to this project, please, let me see I offer to look at your proposals before you submit them. So please do that. All right. There are no other things we want to talk about. I'll go into some things that I wanted to get to today. So let me share my screen again. So this is actually something interesting I found, a paper on phenotype switching in agent-based models of biological tissue. So we've talked about agent-based models. I think in the past we've talked about cellular automata, which are similar types of models. It basically allows you to model things as like a set of cells on a grid or in some sort of topology, and then run a simulation where you have neighboring cells. Depending on the state of those cells, you can you see changes over time. So in a cellular automata, for example, the state of the neighbors influences the state of the central cell, and depending on over time, those you get these global changes that occur over large areas. Agent-based models are an instance of this, where you have these discrete models, you have agents as the cells, and then you have the agents of neighbors, and the state of the neighbors influences the actual the global state of the model, sometimes the local state too. In this case, they're looking at something called phenotype switching, which is something you see in cells. Sometimes you see this in bacterial cells, where you have different types of phenotypic states, and depending on things in the environment, they'll switch their state from one state to another. And it's usually some sort of trigger event like stress, or like a stimulus that they encounter. And so this is an important thing. Sometimes you see this as well in development with different cell states, but this is a term they use in the literature, phenotype switching. So they're modeling this using agent-based models of tissue. So they talk about agent-based models as being an important tool for understanding spatial heterogeneity, which is true because these grids are supposed to be spatially distinct. So, one end of the grid is different from the other end of the grid spatially. You can have things that propagate across the grid and, over time, and you can actually measure that as spatial diffusion or something like that. So the resolution a modeler can achieve in these regards, which are using agent-based models, is unrivaled by other approaches. However, this comes at a steep computational cost, limiting either the scale of such models or the ability to explore, parameterize, analyze, and apply them. when the models involve molecular level dynamics, especially cell level dynamics. So this is where your agent-based models have molecular level resolution, and they're talking about, well, they talk about cell-specific dynamics. So sometimes each agent or each cell has details within it, and so you have dynamics like you might have like a gene regulatory network within an agent that has has some thing underneath the hood of this grid. If there's a state that's being generated by that, and you have inputs from its neighboring cells, then that can be a little bit that can govern the dynamics of the grid and its in the output state, but it's not something you can necessarily directly observe. So these are all important points here. We have developed a global method for solving these computationally expensive dynamics, significantly decreasing the computational time without altering behavior of the system. Here we extend this method to the case where cells can switch phenotypes in response to signals in the microenvironment. And this is this case of like in bacterial cells, you'll see this where bacterial cells will be in a culture dish or something, and you have they might have food in the dish and then they might not, and they can switch their phenotype to like a starvation phenotype or some sort of thing where they conserve their metabolism. So there are different switches that like bacterial cells can have that are very much observable in the phenotype, but it's a complex molecular mechanism underneath it that kicks the switch back and forth. We find that the global method in this context preserves the temporal population dynamics and the spatial arrangements of the cells while requiring markedly less simulation time. We thus add a tool for efficiently simulating ABMs that captures key facets of the molecular and cellular dynamics in heterogeneous tissue. So this is using a tissue model. So they're able to do this. They talk about the limits of running agent-based models on a desktop workstation to around 1 million cells. And they talk about this in the context of a tissue. A lot of cubic centimeter of tissue contains 10 to the 8th cells. So 10 to the 6th is, of course, just a little bit smaller than 10 to the 8th. Or actually, so you can't necessarily on a desktop or I guess a laptop or something of that nature simulate an entire cubic centimeter of tissue. So you're just short of that. So you need to run this on like some sort of cluster to really get a reasonable simulation of tissue. So a cubic centimeter isn't a small amount of tissue, but it's, if we're simulating, if we're simulating a C. elegans, we might be able to do something, approximating a full organism. But if you're talking about like the human body or even a small mammal, you wouldn't be able to do very much. So this is where this sort of computational bottleneck exists. You need to develop techniques to reduce the cost of simulating these agent-based models. And so people have proposed ways to speed up the simulation time or some of the tasks involved in simulating agent-based models. So some of these techniques are simulating a small but relevant region of the tissue. So sampling the tissue down to some process that's ongoing at any one time. Of course, that doesn't give us, we don't really know, we can't really fully define those processes, and there are a lot of processes in the tissue that are important. We've talked about the physics of the full tissue in previous meetings, so that, that would sort of preclude a lot of those processes. There's also partitioning the microenvironment into compartments without internal spatial resolution, which means that you basically, give up on your spatial resolution, very fine spatial resolution, and and the trade-off would be that you can basically have these compartments that just assume homogeneity in certain spatial blocks. So that might be useful for some things, but again, like for other things, if you say for example you have cell physics that are operating and you're interested mainly in that process, then you can probably break your simulation down into blocks that are relevant to that. But you also lose the ability to model other things. So that's a problem. So then you can also target specific parameters, and then using Bayesian inference, that's a good method. In this citation 8, we developed and analyzed what we refer to as the global method for handling molecular dynamics. So you can do global sorts of simulations of molecular dynamics. Those are actually good for speeding up the simulation. These are best for agent-based models that require simulating reactions at the cell's surface or intracellular signaling pathways. So we're moving down from the whole cell down to like signaling pathways and things like that. So there are a lot of ways you can do these models. So the traditional way of doing agent-based models, which we call the local method, is to solve a system of ordinary differential equations for every cell at every time step, which is, of course, this sort of full resolution method. The global method reduces the computational time compared to the local method by averaging the molecular state variables across cells in a region of the microenvironment before solving the governing differential equations and applying the results of those cells. So basically, the local method is where you do everything. You do a full sort of simulation of everything. The global method is where you sort of average across different parts of the local, the localities, and you find the things that are sort of, the most important features, and then you put it into a governing set of differential equations and apply that then backwards to the thing, the different regions that you're interested in. So these are two ways to do this. It's really about granularity, I think. And so it just depends on what you're interested in and what you can get away with. So for some processes, this works and some it doesn't. So, there are different types of things you might be interested in, and depending on what those are, if it's apoptosis or proliferation or symmetric division, some of those things you can model using the global method, and others you may need the local method. So they talk about your agent-based models. They give a set of parameters and their default values here, where they're doing actually something. This is the thing they're doing on phenotype switching. So they're doing this with a cell-based ABM. They're doing two types of agents, proliferating and quiescent cells. So proliferating cells are those that divide frequently and expand their population, whereas quiescent cells are just cells that don't divide and just sit there. And proliferating cells proliferate, move, and die at fixed rates given in this table here. Quiescent cells do not proliferate, and their death and movement rates are orders of magnitude slower than those for proliferating cells. So this is where the kinetics are much less prevalent. And so you can see right away that you have one set of cells that you need to simulate, do these local simulations on, and another population where you could get away with global parameters and be okay. This simulation advances by a Gillespie algorithm, which is an algorithm that people use in molecular simulations. It simulates stochastic processes. Without getting into what that actually, the details of that algorithm, know that this is well validated and it's actually a very common standard algorithm. But it advances by using the Gillespie algorithm, randomly deciding on the next time step given the sum of all rates in the model. This one event for that time step is chosen randomly with the weights given to each event for each agent based on that rate of the event. And so the model advances forward in time until the next randomly chosen time step moves the simulation past the predetermined end time, in which case no cell events are performed. So this just runs down to where you stop having events in the simulation. So then they simulate the molecular dynamics underlying the cellular dynamics. They're simulating the pharmacokinetic substrate. So the pharmacokinetic parameters involve diffusion, cellular exchange, and intracellular signaling, which is basically a stand-in for cellular metabolism. If you were to model that, you'd have like several parameters that you need to basically model locally to get that. We also include a second substrate, quorum factor, in which some simulations that only undergoes diffusion and cellular exchange. So there are these different levels of sort of representing cellular metabolism. And some of these, of course, are needed for the proliferating cells. Others are sufficient for modeling the quiescent cells. And so they kind of go over their methods. They talk about their different, they use different solvers for this. So in these type of models, you're using equation solvers for differential equations. And there are different methods for that. So there's, you know, the internal ordinary differential equation, there's cellular exchange, there's diffusion, there's pharmacokinetics, and people are, you know, so some of these methods span the local and global. Others are local and global. They're, you know, like the diffusion, the LOD method is good for local, and then maybe a hybrid, and then the matrix exponential is good for global. So sometimes the method has to be changed depending on the process. Cellular exchange, you can use a matrix exponential for both local and global, and a direct Euler for the internal ODE for both local and global. So they kind of break that down into how you do that. Then you can coarse grain the microenvironment. So both the global and hybrid methods, hybrid is basically global local in the same model, require partitioning the microenvironment into regions. That is, each lattice site is assigned to a single region. The average of the molecular state variables within each region will be used to solve the differential equations rather than the specific concentrations at each lattice and cell. So what they're doing again is they're doing this global method of averaging out the state variables, using it for a region of this agent-based model, and then applying it to the cells in that region instead of calculating the specific concentration for each cell and lattice site. The choice of regions can be made in any way, which means that, you know, this is it's kind of guesswork, like we were talking about with cell segmentation. Sometimes you have to just say, this is the boundary that I'm defining, and this is my reason, and then that's it. And then, you know, you just apply your method to see how well it works. That's just the way it goes sometimes in modeling. In this work, we shall make use of two choices of regions depending on the location of vasculature in the simulation. For the majority of our work, we shall assume that the vasculature is located at the bottom of the microenvironment. So this is where they're looking at 2 and 3D simulations. So they're actually looking at a 2-dimensional model and a 3-dimensional model. And when they say the bottom of their microenvironment, in the 2D simulations, they have an XY space that where the Y axis is actually the depth. And so the Y min, which are the cells at the bottom of the Y axis, is the bottom of the microenvironment. And in 3D simulations, you have maybe like a cube or a sphere, and that the bottom turns into the Z axis, and the minimal values are the Z axis. So it's like a top-down model with, you know, if you have a cube, you have a depth from top to bottom. The XY, it's usually some gradient where you don't really have a top to bottom. It's, I guess if you plot the bivariate graph, X and Y is at the bottom, but Z is at the bottom in a 3-dimensional graph. So it doesn't really matter, but the point is that you have more spatial resolution in the 3D model. So you basically can have the spatial distribution, the spatial segregation of things, and they can grow across the grid or like the cube or sphere or whatever it is. Under this vasculature assumption, regions will be layers stacked on top of one another, each 1 cell thick. So this is where you have this vasculature, you're building regions on top of one another. In each layer is this layer of vasculature or this layer of the tissue. And so we shall also consider the case in which vasculature surrounds the growing cells. So you can assume that there's vasculature within or you know, around these units and these squares within the model. In this case, regions will be chosen as concentric annually around the center of the microenvironment. So you can define regions as these concentric circles or rings, and you can do this in 2 dimensions, or for 3 dimensions, you can use concentric spherical shells. So you can use different types of shapes to form boundaries within these models. And these are usually these are often used for diffusion of things in the model. So each cell is each square is a cell, and then things diffuse across those cells. And so you have to have diffusion, sort of a limiting distance for your diffusion. So usually people will use a center, or they'll use a sphere or a circle with a center of diffusion, and then things decay linearly from that. So that's what they're talking about here. Then they go through the pharmacokinetics, and that's something that I'm going to leave alone. But this is a schematic comparison of the 2 methods, where you basically have the local and the global. You have the the pharmacokinetic pharmacokinetics, where you have the local versus the global. You have the diffusion dynamics, so you have diffusion, in maybe one direction versus multiple directions. So the global, again, is averaging these things. So you have one arrow coming out the bottom of the pharmacokinetic dynamics model, whereas in the local, you have one for each row. So you can, or for each column, I'm sorry. So you have the arrows, multiple arrows coming down. So there are multiple states here versus in the global, where you have one average state. Diffusion dynamics is the same story, where you have a lot of local interactions versus one set of global interactions. Exchange dynamics is where you have exchange between the cells. In this case, for the local method, you have very local exchange. For the global, you have an exchange per row. So it goes across the entire row and just averaged across that. And we talked about how these are just levels of tissue that go, from bottom to top. And then intracellular dynamics, you have, each row is averaged versus having each cell, things going on in each cell. And they can be different, there's massive, maximal heterogeneity there. So that's basically what it looks like in this model. So they have these different things. They have different strategies for, summarizing things with parameters, or having, specific things for specific cells.
Message right into email.
So this is all. So I
I
guess the results come out as quiescence is an intracellular threshold.
Why can't you just do
So
what I asked you to do?
some of these things are, they kind of agree with some of the. Okay, so for this one, the global method agrees with the local method. So you can see that for some of these results, you get proliferating cells, quiescent cells. You get things that agree across the different methods. So that's good, because we don't want global methods averaging to deviate too much from our local methods. The point of using a global method is to have averages that are, consistent with things that you would get from having maximal resolution. So that's good. But quiescence is an intracellular threshold. Basically, to show the accuracy and speed of the global method, we begin using both the local and global methods to simulate a growing group of cells on a 100 by 100 lattice. So they use this type of method to look at some of these. They look at quiescence in the cells, and they want to see if these things are consistent. And it turns out that they're consistent. So, okay. So, yeah, so then they look at chemotaxis. So to test the validity of the global method in a context where cells can move along gradients at the molecular level, we allow the quiescent cells, which we tested up here, to chemotax, which means to follow chemical gradients. And in this case, they're letting the cells follow an oxygen gradient. So chemotax means to follow a gradient to some maximal value. So, when insects follow a chemical path or a chemical trail of pheromones, that's chemotaxis. But usually we talk about the single cell level. Across all of our metrics, we continue to see agreement between the 2 methods. This includes the quiescent compartment migrating en masse towards the blood vessel at the bottom, and that's figure 4F. So that's actually this one here, where they break this down into these different layers, and so they're following this gradient up and down the model. And again, they're able to look at this in terms of compartments, and they're able to observe this kind of migration. And so they can actually disagree. The local method here agrees with the global method as well. And so they're able to actually do this again for tumor microenvironments using this quorum factor. And so this is an example in 3D of this agent-based model where you have the red and the blue. So the microenvironment, there are differences in the microenvironment. This is the spherical example, but it basically works like the 2D example here, where you have layers that go up and down, and there's migration within the model. So they have a discussion about this. So we've found that the global method for simulating ABMs is sufficiently robust to handle discrete effects of the molecular scale, or of the molecular scale on the cellular scale. So we want to simulate the molecular dynamics and look at what the effect is on cells and switching cell state. So we can do this. We can do this for this global method and this local method. We can capture the process of chemotaxis along a gradient. And we can also do this to look at cell fate decisions, which are these switches, these phenotype switches. We can extend that to how a cell decides to make a change in fate. In other words, a change between like a stem cell and a differentiated cell, or two different types of differentiated cell. So this is a nice paper kind of describing in excruciating detail how these processes work and how we might model them. So, okay, that's all I have for that. And I'll put that paper in the Slack if people are interested in reading it. But I think that's a nice overview of models, like using a biological model to solve some of these problems. And we talked about cell segmentation. So in cell segmentation, we're taking real world data, we're breaking it down into parts that we can analyze. And in this case, we're taking those parts, we're simulating them, doing sort of a backwards approach to understanding what they're doing. So we could take our segmented data, put it into this model, use it to calibrate the parameters, and then simulate things forward to observe things that we might not be able to observe under a microscope, or get like more general trends in the biology. Yeah, I'd like to talk about a paper on butterfly eye spots and morphogenesis and using convolutional neural networks. This paper should be a treat. So the title of this paper is Detection and Measurement of Butterfly Eye Spot and Spot Patterns Using Convolutional Neural Networks. And this is a group out of Portugal and Singapore. So this was published recently in PLOS One. So the abstract reads, butterflies are increasingly becoming model insects where basic questions surrounding the diversity of their color patterns are being investigated. Some of these color patterns consist of simple spots and eye spots. So you can see that there's the Wikipedia stuff on eye spot. Eye spots are a form of mimicry. So the butterfly wing mimics different eyes that might be in nature, mimicking another organism like a predator. And they actually, in general, they have very colorful wings. And these wing patterns, sometimes they're mimicry, sometimes they're other things. And so this is attributed to a pattern-forming biological process of morphogenesis. So this is controlled by a number of genes in embryonic development, such as engrailed, distalis, hedgehog, antennapedia, and notch. So these are all signaling pathways that interact in the formation of these. So this is something that you see early on in the organism's development. In the caterpillar, you have these imaginal discs that have these sorts of genes being expressed, and then during pupation, these wings form, and then you get the butterfly with colorful wings. So they kind of go through things that aren't related to butterflies in this article. But in the main article here, let's go back now that we know what eye spots are and other types of spots on the wings. We can talk about maybe some of the things they're doing to analyze them. So to accelerate the pace of research surrounding these discrete and circular pattern elements, we train distinct convolutional neural networks, or CNNs, for detection and measurement of the butterfly spots and eye spots on digital images of butterfly wings. We compared the automatically detected and segmented eye spot areas with those that are manually annotated. So they're doing automatic detection of them, which means that you can take an algorithm and try to find patterns, but you don't know exactly what those are. You might find a lot of alternate variation around pattern formation, might find things that are spurious patterns that maybe we don't see with our eyes. And indeed, the patterns themselves aren't really determined by any cognitive mechanism. They're determined by the expression of these genes in these circuits during development, and especially during pupation. So you get this, basically you want to be able to detect things that may be interesting in terms of pattern formation. But CNNs are good at that, but sometimes they're not trained on the things we might see in, the things that are actually patterns. I guess that's the way I'd put it. In any case, you have this manual annotation of things that we've seen in other butterflies that might be of interest, as opposed to just something that maybe repeats, or that's a pattern in the wing. On the other hand, maybe there are patterns that CNNs can uncover that human observers can't. So there are a lot of opportunities for human-machine cooperation here. We then compared the automatically detected and segmented eye spot areas with those manually annotated. These methods were able to identify and distinguish marginal eye spots from spots. So they're spots, and then there's spots that look like an eye. So if you go back to our mimicry example, we have spots here, things that are of a different color. We have this red spot here versus, say, like an eye spot here with looks like an eye. It looks like it has the white of an eye and then the pupil. So you see some of these down here. I guess these would be considered eye spots, but this main thing here wouldn't be an eye spot. It would be a spot. This would be a spot, and so forth. So there are a lot of patterns to detect on a wing. These methods were able to identify and distinguish marginal eye spots from spots, as well as distinguish these patterns from less symmetrical patches of color. So these are like patches, that have this kind of, this might be a symmetrical patch. This might be a less symmetrical patch. Just examples. If you zoom in, you can find them. In any case, there's a lot in there that we need to look at and understand and quantify. In addition, the measurements of an eye spot central area and surrounding rings were comparable with manual measurements. So people were able to measure them, detect them by a human eye. Expert recognition is usually people who have looked at many, many butterfly eye spots or many, many butterfly wings, and they know what these features should look like across butterflies. And so we also have measurements of those features. And so being able to match up with those measurements is actually pretty good for the CNN, from the CNN's perspective, because it means that it's locking on to patterns that at least human experts have found interesting. These CNNs offer improvement of eye spot detection and measurements relative to previous methods, because it is not necessary to mathematically define the features of interest. So you don't need to mathematically define these features. And this really kind of stands in contrast to some of the differential equations we use to model reaction diffusion and other types of pattern formation. In that, you don't need to mathematically define the features, but you need to mathematically define the process. And so in this case, we're just looking for patterns. We're using a statistical method to find patterns that are common across the images. So this is actually something that stands in contrast not just to other types of measurement methods, but to other types of ways of looking at morphogenesis. all that is needed is to point out the images that have those features to train the CNN. So basically, you can train the CNN on things that you know are patterns, patches, eye spots, and give that information to the CNN, and it can extract them out of the data, even when maybe they're not immediately apparent to the human expert. So they talk about eye spots being salient color pattern stimuli with multiple rings of contrasting colors that mimic vertebrate eyes. They are used by a variety of animals primarily to intimidate or startle predators or deflect predator attacks to dispensable areas of the body. So this is just a way to fool predators basically and keep them from killing the butterfly. So it increases the butterfly's fitness. Eye spots have been studied primarily in the Lepidoptera, where different modes of defense are found in different species, and where eye spots are also used in sexual signaling. So we saw examples of the eye spots in different species, aside from butterflies, and this has mainly been studied in butterflies, however. Eye spots in nymphalid butterflies have a single origin, this evolutionary origin, and they may have evolved from a simpler set of pattern element spots. So, there are different types of patterns that came together in morphogenesis to form these eye spots, and this was, of course, there was a fitness imperative. So it came from precursor spots that are common on butterfly wings. Spots themselves are simple patches of color contrasting against the background color of the wing. It is unclear how many times spots have evolved independently in butterflies, meaning they have multiple origins in phylogeny, meaning that, they're just basically artifacts of pattern formation. Spots appear because that's how butterfly wings are patterned. These gene expression networks express things maybe more or less stochastically, and this has multiple phylogenetic origins. However, eye spots, being very specialized and having a fitness imperative, maybe evolved once in a single lineage and then diversified. Okay, and then, so we don't really know what its exact ecological function is, but we have a pretty good idea that eye spots are have a high fitness. The accurate measurement of spots and eye spots has become a routine task for researchers. Different size eye spots are found in males and females, so there's sexual dimorphism there. So this means that there's probably plays a role in sexual signaling. And there's also differences in eye spot size between the dry and wet seasons. So there's phenotypic plasticity in the same species. There are also different predator guilds that form, so the eye spots have to change. And so these eye spots aren't constant across the lifespan. They change seasonally, but they also are they originate in pupation and even further in development. So measuring spots and eye spots has also become important for researchers who explore mechanistic questions. So you can ask all sorts of ecologically oriented questions. Then they decided, that you can actually measure these things with calipers or with some other quantitative method, but you need to classify them to know kind of, if it's a spot or an eye spot, because there's a lot of variation in the data. So you can use, people have in the past used SVM classifiers to detect sort of which spots are in which category, and then they build a circular ring and they measure a 1D Ho transformation for circle detection. So you have these circular rings that you can derive for different types of spot, and then you can use this transform to detect the circle. And so that was the state of the art before they applied this convolutional neural network approach. And so this is a very common approach in deep learning, and they're able to do this. And of course, you need a lot of training data for this. So they were able to use a lot of training data from measurement data that exists. There are all sorts of different ways to do this. A popular approach for implementing CNNs is to segment the images into two classes. And in this case, they're using the nucleus in the background, and then they're able to Yeah, so since semantic segmentation, which we talked about earlier in the meeting, cannot separate touching or overlapping nuclei, post-processing approaches such as watershed or H-minima transform are commonly used. So this is an interesting point with respect to some of the stuff we were talking about earlier. In this case, we have these classes, and sometimes the nuclei are touching or overlapping, as we see in single cells. Sometimes if they're dividing, you have this problem. And using something like instance segmentation over semantic segmentation can improve performance. However, you can also use things like watershed or H-minima transform to do the same thing in a post hoc manner. So this is post hoc to the data collection. So you can actually, maybe use a watershed algorithm and then a CNN. So you get you do one form of segmentation and then plug that into the CNN as sort of a supervisory data set or some combination of that. We've done this with the Bradly where we did some algorithms on that data, which is different from, say, this data or the single cell data that we've talked about in embryos. Watershed on the Bradly data did pretty poorly, and so we had to come up with a technique that would allow us to approach that specific problem. So in this case, they're suggesting that watershed is actually a pretty good method, especially for post-processing of the data. I think that it's interesting that these methods aren't universal. In other words, it really depends on the problem you're trying to address. So they get into some of these things about convolutional neural nets. They talk about fully convolutional networks, which are a type of CNN composed of only of convolutional layers and without fully connected layers, can be used to look at nuclei segmentation maps, and then following that up with watershed post-processing. So in this case, they use nuclei segmentation using the FCN, and then they do watershed post-processing. In this paper 27, they use FCN to predict a distance map to centroids and boundaries of nuclei. So they use this FCN method to get the centroids and then determine the boundaries of nuclei as a distance function from the centroid. So there are different ways you can do these kind of segmentation procedures. Instance segmentation methods such as mask, RCNN, jointly detect and segment, thus solving the splitting problem, which is where you have splitting of cells that are single cells and so forth. So so this solves this problem. It could solve it in butterfly spots, or it could solve it in single cells in an embryo. So in this type of image segmentation, however, the images of the cell nuclei and cell background are similar, which is not the case in the images of our diverse species of butterflies. So in the cases where they're using this mask RCNN, they're talking about, some of these limitations. But in this specific case, you have a problem where you can't really separate out the nucleus from the background. You have to treat them sort of in a different way because you can't really separate them out very easily. So they use CNN for both eye spot detection and measurements. They first use a data set of images of different species of butterfly with a variety of spots and eye spots for training and testing 3 state-of-the-art object detection methods. Then we test the best CNN to detect eye spots in a different image data set, consisting of images of many individuals of single species. So what they do is they try different CNNs, and then they select the best model to detect eye spots across all types of images of these eye spots. And then in the second image data set, we also train and test a new CNN to segment and measure different eye spot areas. So this is the materials and methods where they talk about the data set that they're using. They're using a pretty decent sized data set of 4,707 butterfly images. It's actually not that huge, but it's, it depends on how much variation there is in it. The key thing here is that the images, they're like 4,700 images, but you have almost 20,000 spots and 20,000 eye spots. 15,000 1,000 to 20,000. So that's really the N you're working with. And so this is enough to train the model. So they're able then to detect these patterns. So this is an example of the input data. You have these different spots and eye spots that are sort of isolated on the wing, and then the algorithm has to segment out these parts from these images. And so this is an example, figure 2, illustration of ground truth. And then the white are the regions we want to measure for. So we have these eye spots, and you can see sometimes they're pretty blurry, sometimes they're hard to separate from the background, and you have to create these masks which show the area you want to segment out. So the white in the background is this white spot, and these brown spots back here, and that's the black, and then the white is the area that's the target, which is the eye spot itself. So you can see that they create these masks that give you a ground truth. Then they use, let's see, in this one, they use two methods. They use eye spot detection, which is just this general eye spot detection. Then they use UNet as a form of eye spot measurement. So they have the detection, then they have this extraction and resizing of each detection. Then they have the measurement through UNet, and then this resizes it to the original using these masks. And they go through a lot of technical detail on some of the details of their model, but basically they're using CNN and then they're using UNet for these different things. So they have the code for all this in a GitHub repository. There involves YOLO, which is you only look once, RetinaNet, EfficientDet, and UNet CNNs. And we've worked with a number of these models, so this is something that we have code for as well for embryos and segmenting cells and embryos. So we implemented 2 versions of UNet that can provide us with the same set of measurements, namely area of the white center and the surrounding color rings. So one segment's only the 2 color rings from the rest of the image. That's their 2-class criterion. And the other segment's center color rings and background, which is the 3-class criterion. So basically you have the color rings, the background, and the center all as different classes versus the background and the center is 1 class and then the ring is the 2nd class. So they use the categorical cross entropy cost function to determine, which parts are which. They use the unweighted version of this cost function, which is more commonly used, as well as a weighted version, which includes class weights. So all the code is in this GitHub repository. And so they have a number of measurements that they use for quality control. They look at the pattern element. A pattern element is considered a true positive if the intersection over union between its bounding box and any ground truth bounding box. So we talked about bounding boxes with respect to single cells, but basically a bounding box is something you define as an element around. You find like the edges, and then you sort of infer a bounding box around that based on the edges. It might be the distance from a centroid or whatever. But then you can actually use that against the ground truth. And the ground truth, of course, is the mask that we saw. And so you can combine those two, looking at the area of intersection over the area of union. And so intersection is where the 2 sets intersect versus the union where they're the same. And so you get this measurement of IOU, which is this intersection over union, and that gives you an indication of the ground truth. So, we can do it that way. We can get a score for its reliability and its true positive, the nature of a true positive. So we want to throw out the false positives, basically. So this is, again, an example from the data set. The different methods and their performance. And then we get our performance here on eye spots, marginal eye spots, and all. And the performance is pretty decent. It's not perfect, but it's decent. So this is an example of the masks and the ground truth. And then the actual segmentation of the eye spots. And so this is, I think this stands well into contrast with a lot of the segmentation algorithms we use for cells and embryo, it's a different type of system, different dynamics, but it's also, it does give us some interesting parallels to consider. The other thing is that it stands in contrast to some of the models of morphogenesis. So we've talked about models of morphogenesis using differential equations. We've talked about models of morphogenesis using physical attributes like the physics of cells, and we've talked about pattern formation using genetic regulatory networks. So a lot of these genes that are involved in pattern formation, like spot formation and striping and things like that, segmentation, those are all, you can build models of genetic regulatory networks that produce these patterns. And so the question is, what is the relationship here? We could model, say, for example, a GRN or a differential equation with noise or with environmental inputs that allow us to model some of these ecological variations. Could we generate a data set that then this algorithm could detect patterns versus non-patterns? And then you're basically we're asking two questions there. One is that can your model of a genetic regulatory network produce things that can be recovered by convolutional neural network that's trained on detecting patterns in actual biological phenotypes? And then two, can your model of the phenotype or the genetic regulatory network expressing a phenotype, can that match what you see in nature? Can that match the variation in nature? Can that match the patterns that are produced in nature? So there's some really interesting things you could put together from this. Again, I'll post the paper in the Slack. I think that, if you're interested in CNNs, if you're interested in pattern formation, this would be a really interesting thing to follow up on. So thank you for your attention, and I hope you learned something. Okay. Any questions or comments before I think we'll wrap it up for today. Okay. So again, if you have any questions about GSoC or any other projects we're doing or anything of interest, let me know. And yeah, I look forward to seeing progress for next week. Sushmanth?
Yeah, I will definitely let you know next week. I was right now working on proposal only.
Okay.
I will complete the proposal and then I will come up with the code and all. Actually, the work for the GSoC I have started, like segmentation and all for extracting those centroids, volume, etc. I will show you update as soon as possible.
Oh, that's great. Yeah, I look forward to it. Yeah, I think you're making good progress. I wanted to make sure you knew that because, it's just that we want to. We're kind of doing this roadmap on the fly because, we spend time on the project in the summers and then it kind of grows fallow in the winter time. So it's like, we don't have a full-time focus on it necessarily, but that's okay. We just have to make progress where we can.
Right now, should I work on proposal or should I go with the code and all, right? It's like, so there is no time actually. From next week onwards, for me, exams have been starting.
Yeah.
Before that, I want to work on proposal. That's the reason I was giving more time on proposal.
Yeah, I think at this stage, the proposal is the most important thing. I mean, the code you'll be doing in the summer, Google Summer Code. So, that's where that would come in. Like, you don't want to do the whole thing now. You want to just give enough so that you can make some maybe figures and tell us. I think at this point, you've got certainly enough detail for a proposal. It's not a problem.
Okay.
Right.
I will work on proposal and I will, by next week. After that, I have pretty confused week, like exams and all are there for me. I'll be busy with that. Before that, I will complete my proposal and I will submit to you for a review.
Okay, it sounds good. All right. Thanks. Have a good week, everyone.